So, students, today you'll be studying about the Sycon morphology and the body wall. Okay. So, here, coming to the BSc first year zoology, BSc first semester zoology, you'll be studying about the Sycon morphology and its body wall, how the body wall will be. Okay. So, this you are going to study today. Now, if you take the Sycon, okay, Sycon, it is also called as the urn sponge, URN, okay, urn sponge, because of its shape, because the urn is nothing but pot-like, okay. So, urn sponge, and it is also known as the crown sponge, because the crown is present, which is made up of straight spicules, which are present, straight and long spicules that are present on the anterior region of the Sycon. So this straight and long spicules which are present at the top looks like a little crown. So it is also called as the crown sponge. Okay. So if you take Scypha, so Scypha or Sycon we call it as. So it belongs to phylum Porifera, class Calcarea, order Heterocilla, and the family is Cytidae, and the genus is Sycon or Scypha. Okay. So now coming to the class Calcarea. So this Sycon, it is having a calcareous spicules. So only it is coming under the class Calcarea. Okay. So you have to remember this. Now if you see this structure or external morphology, okay. So it is having a urn-like body, okay, urn body, or it is somewhat cylindrical in nature, okay. It is cylindrical. It is having a cylindrical body, and you can see here at the anterior region, you'll find the straight spicules are present. So inside these spicules, you'll find the osculum will be present, okay. And these spicules, they are called as the oscular fringe, okay. And these spicules, they are made up of monoaxon spicules. And if you take this Sycon all over the body, you can see many number of pores are present, okay. And these pores, they are called as the dermal ostia, okay. So Sycon, it is having a cylindrical body, cylinder-like body, okay. And you can see at the anterior region, the oscular fringe is present. So this oscular fringe, they are made up of monoaxon spicules, and this oscular fringe is surrounding the osculum, okay. And on the body surface, you can see many number of small pores are present, which are called as the dermal ostia. And see, here also you can see the spicules. All over the body also you can see the spicules, but these spicules, they are smaller compared to this oscular fringe, okay. And you can see the base is present. So this base will help in the attachment of the Sycon to the substratum, okay. And so during budding and also a bud also will grow, okay. So this is how the Sycon morphology will be. So you have to draw this diagram regarding this morphology of Sycon, okay. So how the body is wash shape or the cylindrical shape. So it is having a height of 2.5 to 7.5 centimeters, okay. So it can grow up to 7.5 centimeters. So they are branched also, okay. And the branch will be attached to the common base, okay. So I was telling the bud, okay. So it is attached to the common base. And if you take the body wall of the Sycon, so body wall of Sycon, it is made up of 2 layers, okay. So these 2 layers, the outer layer, it is made up of flat cell, which is also called as the pinacocyte. And on the body wall, you can see many number of ostias are present. And if you take the inner layer of cells, they have a flagella, and they are also called as the conocytic cell, okay. They are also called as the conocytic cell. Then you'll find many number of spicules are present. So these spicules, they are needle-like spines. So they are the skeleton of the Sycon. And this spicules, they are found embedded in the jelly-like substance, which is also called as the mesoglia. Okay, it is also called as the mesoglia. And some of the spicules, as I told you, they are monoaxon spicules. They are surrounding the osculum, which are also called as the oscular fringe. They are little bit longer compared to the other body spicules. Then on the body of the Sycon, you'll find many number of pores are present. So these pores, they are also called as the ostia or the incurrent pores. So these pores or the ostia, they lead into the channels. So through the ostia, whatever the water that is there, it will enter through ostia, and there is one large single opening, as I told you, that is the osculum. So through this osculum, the water will exit. Okay. So the incurrent water is coming through the ostia, and the excurrent water is going out through the osculum. Okay. So if you take the canal system of the Sycon, so the water will enter through the ostia. From there, it has to go to the central cavity, which is also called as the spongocoel. Okay. So if you take the Sycon body, so it will be usually radially symmetrical. Sometimes it looks asymmetrical also. Okay. So the body is radially symmetrical or asymmetrical. Okay. So if you take the Sycon canal system, okay, Sycon canal system, it is also called as the Syconoid type of canal system. That is grade 2 Syconoid type of canal system is found in the Sycon. Okay. So here, if you take the how the water will enter, so the water will enter through the dermal ostia. Then from the dermal ostia, it will move into the incurrent canal. From the incurrent canal, it will move through the move into the radial canal through prosopyle, and from the radial canal it will move into the spongocoel through apopyle, and from the spongocoel, which is a body cavity, the water will move out through the osculum. Okay, so that is how the way of water will be seen in the canal system of Sycon. Okay, now coming to the TS of body wall of Sycon. Okay, so here the body wall of Sycon, it is usually folded, okay, at regular intervals. Understood? So whatever the folding that is there, so you'll find the invagination and evagination is been formed because of the folding. Okay, so alternate evagination and the invagination occurs. Evagination is nothing but the out pushing of the body wall, and the invagination is nothing but the in pushing of the body wall. Okay, so the Sycon body wall is been folded, and it forms a alternative invagination and the evagination occurring. Okay, so the as I told you, the Sycon, okay, the Sycon, it is having a canal system. So through the ostia, the water will enter. From the ostia, it will and water will enter into the incurrent canal. From the incurrent canal, it will enter through the prosopyle to the radial canal, and from the radial canal, it will enter into the spongocoel through the apopyle. Okay, then from spongocoel, it will enter into the osculum. Okay. So here the spongocoel, it is also called as the body cavity. So it is the other name of the spongocoel, it is also called as the paragastric cavity or gastral cavity. Okay, you can call it as gastral cavity or paragastric cavity. So osculum, it has been provided with the sphincter. Okay, and this sphincter, it is lined by the special type of cell, which is also called as the myocyte. Myocyte are nothing but the muscle cell. Okay, myos means muscle, chitos means cell. So they are one type of muscle cell. So the sphincters are present near the osculum. Why the sphincter should be there? Because whatever the water that is moving out, okay, how much of water has to move out, so that has to be controlled by the sphincters. Okay, so that is why the sphincters are lined by the muscle cell, and it is helping the osculum how much of water has to move out. Okay, so that is how these myocytes are lining the sphincter, and the sphincters are present near the osculum. Okay, and if you take the body wall of the spongocoel, so the spongocoel is nothing but the cavity that is present. So as I told you, the sphingosyle has the regular interval of the evagination of the body wall has been seen. So which is forming the radial canal. Evagination of the body wall will give rise to the finger-like radial canal, and invagination will give rise to the in between the radial canals. Okay, regular interval the evagination is occurring. So in between the evagination, the invagination is there. So this invagination, it is forming the incurrent canal. Okay, invagination will form the incurrent canal, whereas evagination will form the radial canal. Okay, and this radial canal, it has been lined by the flagellated conocytic cell. Okay, radial canal is always lined by the flagellated conocytic cell, and the radial canal, it is connecting the sphingosyle through a small internal ostia, which is also called as the apopyle. Okay, apopyle is also been surrounded by the contractile myocytes, so which is also serving as a sphincter. Okay, so if you take any two radial canal, so in between the radial canal, you'll find the tubular incurrent canals. Okay, so this incurrent canal, usually it will be lined by the pinacocytes. So radial canals are been lined by the conocyte. Conocyte layer is the inner layer. Okay, that is the endoderm, we call it as. Then pinacocytic layer is the outer ectoderm. Okay, so pinacocytic layer is lining the incurrent canal. Then if you take the conocytic layer, it is lining the radial canal. Okay, so you have to remember this. So in between the incurrent canal and the radial canal. Okay, so if the water has entered into the incurrent canal, it has to move to the radial canal. So in between that, it is having a minute pore. Okay, and this pore, it is called as the prosopyle. Okay, prosopyle. So where is the prosopyle? Prosopyle is present in between the incurrent canal and the radial canal. Okay, so you have to remember this. So now this after the radial canal. Okay, from the radial canal, the water has to move into the sphingosyle. Okay, the water has to move into the sphingosyle through the apopyle. Okay, through the apopyle. Then once the water enters into the sphingosyle, it will move into the osculum, and from the osculum it will be thrown out. Okay, so the water will be thrown out. So only water will not move. So along with that, you will find the waste food particles. Okay, and also the whatever the excretory product, all that will be moving out through the osculum. Okay, so that is been seen. Then if you take the body wall microscopically, so here you'll find the single layer of that is the pinacocytic layer, single layer of pinacocytic layer that is found covering the external surface of the body. Okay, then if you take the sphingosyle. Okay, sphingosyle, it is lined by the flattened cells. Okay, so these flattened cells, they are found in the sphingosyle. Okay, then if you take the radial canal, it is lined by the conocytic cell. And if you take the incurrent canal, it is lined by the pinacocytic layer. Okay, so in between pinacocytic layer and the conocytic layer, there is a gelatinous matrix. So this gelatinous matrix is composed of the spicules and the amoebocytes. Okay, so I can see here in this diagram. In this diagram, see the body wall. Okay, so here this is the osculum. Oscular fringes are there. So these oscular fringes, they are made up of the monoaxon spicule. And then if you, this is the osculum, and see the body wall, how it has been having the invagination and the evagination. So this is the evagination, this is the invagination, evagination, invagination. So alternating evagination and the invagination is occurring. So evagination, it is forming the radial canal. Invagination is forming the incurrent canal. Okay, so in between the evagination and the that is the incurrent canal and the radial canal, you will find the prosopyle will be present, and in between the radial canal and the sphingosyle, there will be apopyle present. Okay, and the pinacoderm layer and the conoderm layer you can find, and you can see the spicules here. So like this, if you dissect the Sycon, you will find like this. Okay, the wall of the Sycon will be present like this. And see the apopyle, how the apopyle will be. Okay, so here apopyle, it is the central, that is the pore that is there, and surrounding that you can see the myocytes are present with the mesenchyme. Okay, so the mesenchyme is there. So then if you take here the dermal ostia, so here the dermal ostia. Okay, so this dermal ostia, it is also middle region. Okay, middle region, you can see the pore like structure. Okay, so this is the pore like structure. Surrounding that you can see the myocytes are present. Okay, so like this the dermal ostia is there. So this is the enlarged diagram, that is the section of the body wall which has been shown. Okay, so here you can see here the radial canal. Okay, so this is the part of the radial canal. Okay, this is the radial canal and this is the incurrent canal. Invagination is incurrent canal and evagination is radial canal. And you can see here this radial canal. Okay, this is the radial canal. So radial canal is lined by what? Conocytic layer. Okay, you can see the conocytic layer, flagellated cells, we call it as conocytic layer. And see here the incurrent canal, it is lined by the that is the pinacocytic layer. Pinacocytic layers are having a flat pinacocytes. Okay, flattened pinacocyte, you can see here. Incurrent canal is lined by the pinacocytic layer, that is the flat cells. And the radial canal, it is lined by the flagellated cells. Okay. And in between, see here, in between this pinacocyte and the conocyte, this is the mesoglia. And in the mesoglia, you can see the amoebocytes are present. Okay. And you can see the spicules are present. These spicules are of a different type. Okay. Some are monoaxon spicule, some are triaxon spicule, triates, okay, triradiates, or even the tetraradiates are also been seen. Okay, you can see these are all the monoaxon spicule. Okay, so like this you'll find the mesoglia, it is been present, and you can see even one small larvae is also there, that is the amphiblastula larvae. Okay, so this is seen in the mesoglia. Okay, it is found in the mesoglia. So worm is also there in the mesoglia. And see here, in between the radial canal, okay. So this is the incurrent canal and the radial canal. So in between the incurrent canal and the radial canal, this is called as the prosopyle. Okay, the pore that is present is called as the prosopyle. And from radial canal to spongocoel, so this is the spongocoel. You can see here the apopyle. Okay, you can see the apopyle. So this is the outer, that is the water present here. This is the exterior part. This is the spongocoel. Okay, that is the cavity present, body cavity that is present. So like this you can see. Okay, so this is the conoderm layer present lining the radial canal, conocytic layer, conoderm, and the incurrent canal, it is lined by the pinacoderm. Okay, so like this you can see. And here you can see the ostium. Okay, ostium is there. So water, how it will enter? Water will enter through the ostium, then to the incurrent canal, then prosopyle, then radial canal, then apopyle, then the spongocoel. Okay, so that is how the water will enter. So this diagram is very important for the section of the body wall. Okay, so this is important. So now if you take the pinacocyte, okay, so pinacocyte, they are covering the outer surface and they line all the internal canals, that is the incurrent canal. Okay, but the radial canal is lined by the conocytes. Okay, so pinacocyte, usually they are syncytial epithelial cells. Okay, syncytial means what? That is the cells having 2 or 3 nucleus. Okay, then that is the external lining of the Sycon is also done by the pinacocyte. Okay, the outer body surface is been lined by the pinacocyte, which we call that as a exopinacocytes. Okay, and the internal lining of the pinacocyte, that is covering the spongocoel, which we call that as a endopinacocytes. Okay, now these pinacocytes are large flattened scale like cells which are held together by the thin margins. Okay, and they have a centrally placed nucleus. Okay, and they are highly contractile, and they increase or decrease based on the surface area of the sponge body. Okay, then if you take the conocyte, so conocytes are also called as the collar cells or the gastral cells. So they have a long whip-like flagellum, and each conocyte has a round oval body, and it possesses a single nucleus with one or more vacuoles, food vacuoles, reserve food particles. All these are present in the cytoplasm of the conocyte, and the free end of the cell body has a longer flagellum, and base of the flagellum is surrounded by the contractile transparent collar-like outgrowth of the cytoplasm, and the collars have the filtering mechanism for trapping and engulfing the food particle. Okay, here, so like this it has been seen, and the flagella that is there. Okay, so flagella of the conocyte will help in pulling the water into the collar. Okay, and also the food particles along with the water will come to the will be entering into the food vacuole. So the conocytes have the flagella. The beating of the flagella will make the water to direct towards the towards the apopyle, so that it can enter into the spongocyte. Okay, so apopyle are excurrent pores. Understood? Or we also call it as the internal ostia. Okay, so that is how it is helping. So you can see here, this is one of the electronic microscope structure of the conocyte. You can see here this conocyte, how it is. Okay, so you can see the cytoplasm. Inside the cytoplasm, you can see a nucleus is present. Okay, and you can see at the base the protoplasmic strands are there. Then you'll find the food vacuoles. Okay, all these are present. Then here you can see the flagellum. A single flagellum is present, and it has been starting from the basal granule. From the basal granule is connected with the rhizoplast to the blepharoplast. Okay, and you can see this is a collared cell, as I told you. Okay, so they have the collars here. Understood? So these have these collars. Collar is made up of the microvilli and the micro fibrils. Okay, so you can see here this is the microvilli, and the microfibrils are present, which is forming the collar in the conocytic cell. Conocyte. Okay, conocyte cell. And how this that is the microvilli and the flagella are microfibrils are arranged. You can see this is the microfibril, and this is the microvilli. Okay, these are the microvilli, and this is the microfibrils. This is how it will be present. Okay, yeah. So no need of drawing this. This diagram is enough. Okay, so that is about the conocytes. Then if you take the mesoglia, so mesoglia, it is present in between the pinacocyte and the conocytic layer. So it is more in quantity, and also many number of spicules and amoeboid cells are present in the mesoglia. So this different types of the cells are there. Okay, so amoebocytes, they give rise to the different types of cells. Understood? Whenever it is amoebocytes, they can just become a different type of cells. Okay, whenever it is required. So amoeba, so here amoebocytes. Okay, amoebocytes, they can change their function based on the whatever the Sycon requires. Okay, so the amoebocytes can become colonocytes. Understood? And when they become colonocyte, they develop a slender branching pseudopodia, and they unite to form a syncytial units. Okay, then the amoebocytes, they become chromocytes. So if they become chromocyte, they will have a pigment, and the pseudopodia will be lobose pseudopodia. Okay, then trophocytes are there. So these trophocytes are the nourishing amoebocytes. They will have the food particles, and this can help in digestion and distribution of the nutrition. Okay, Then the thesocytes. Thesocytes are the storage amoebocytes. So they also have the lobose pseudopodia and are filled with the reserve food material. They keep the reserve food in the thesocytes. Okay, then archaeocytes are there. So these archaeocytes are undifferentiated embryonic cells. They later form the sperm or the ova. And these archaeocytes have the blunt pseudopodia with a large nucleus. And if you take the scleroblast cells, so they are one type of amoebocytes. They secrete the spicules, spongin fiber of the skeleton. So these amoebocytes, if they become calcoblast, they form the calcareous spicules. If they become silicoblast, they secrete the siliceous spicules. And the spongioblast, they form the spongin fiber. So this calcoblast, silicoblast, spongioblast, all are amoebocytes. Okay. These amoebocytes, they can differentiate themselves into the calcoblast, silicoblast, or the spongioblast to secrete the spicules and the spongin fiber. Then desmocytes, so these are elongated cells. They are found in the outer cortex. So they give the strength to the cortex region and also for cortex contraction. Then myocytes, so they are elongated fusiform contractile cells. Just like how the smooth muscle works, the myocytes will help. So these myocytes are found in the sphincters, that is near the osculum and the apopyle. So they will help in regulating the movement of water. Okay. When the water enters into the body and when the water is moving out of the body, so these myocytes will regulate. So you can see here, this is the pinacocyte. Okay, flattened cells. Then second one is this is myocyte. Then this is the porocyte, prosopyle. Prosopyle is lined by the porocyte. Okay, so here one canal will be present. Then this is the thesocyte, and this is the conocyte, and this is the scleroblast cell. Okay, and this is the archaeocyte. This is the colonocyte. Okay, so this is the colonocyte, and this is the conocyte. Conocyte with the flagellum. So these are the cells you'll find that are found in the Sycon. So if you take the skeleton that is present in the Sycon, so all the spicules that are there, they are called as a skeleton, and they are made up of calcareous calcium carbonate. So they are called as the calcareous spicules. So they'll be arranged in the that is in the mesoglea, and they'll be present on the body of the Sycon and near the osculum and all. Okay, so you can see the monoaxon spicules are there. So this is the monoaxon spicule. They are large, straight, and only one ray is present, and they are arranged in a circlet manner near the osculum, so forming the oscular fringe. Then oxyate spicules, they are also monoaxon spicule, but they will have a spear or club-like structure, and they are found in the dermal cortex. Okay, and then if you take the triradiate, so this is the triradiate. They have the 3 rays, and they are present near the flagellated canal, that is the radial canal, with one or more ray pointed towards the distal end of the canal. Then tetraradiate, so these are the tetraradiate. Okay, so these are found in the spongocele. Okay, tetraaxon or tetraradiate. So all these are the different types of spicules which are present. So here hexaaxon is there. Okay, these spicules are present in the scypha or the Sycon. So these are the spicules you have to write. So this forms the skeleton of the scypha. So where it will be present? It will be present in the embedded in the mesoglial matrix. Thank you. So this is about the Sycon. You have to remember this and write. Thank you.